The real science of Jurassic Park. Can we really bring back dinosaurs with genetic engineering? Let's start off with some basic facts. Dinosaurs vanished 66 million years ago. The practical half-life of DNA is around 521 years. Even under ideal conditions, usable genetic information crumbles to dust on the timescales of millions of years. So, is Jurassic Park a lovely lie, or does modern biotech give us a different way in? Welcome back to the Real Science of Sci-Fi. Today we're going to interrogate the math, molecules and science behind de-extinction. We'll crunch the numbers, kill a few myths and explore some real routes scientists are using right now to clone extinct organisms from DNA. So, DNA. DNA is chemically fragile. Water breaks down the phosphodiester bonds of DNA. Heat accelerates this process. Oxygen and background radiation add additional random breaks in the DNA strand, and cytosine deaminates into uracil, causing CT errors over time. This all happens over hundreds of years in ideal conditions. As we said, the half-life of DNA is 521 years. This means that after every 521 years, about half of the bonds in a given DNA strand have snapped. As a rule of thumb, after about 7 million years, even under really good conditions, strands of DNA are now so short and damaged that true genes are gone. The last non-avian dinosaurs died out 66 million years ago. That's 10 times longer than the theoretical limit that DNA would have survived. So it's not looking good so far for Jurassic Park. To assemble a genome, we don't need one intact chromosome. You need many tiny fragments that all overlap. Modern labs typically aim for read lengths of 30 to 100 base pairs and a target 30 times coverage for a high-quality genome. For a 3 billion base pair genome, something akin to an elephant, that's 93 billion bases of sequence data. When your average fragment length collapses to dust and miscoding lesions dominate, assembly stops being science and becomes wishful thinking. The oldest animal DNA recovered from a specimen is around 1.2 million years old. This was a mammoth tooth from Siberia. Impressive, but still 50 times younger than the dinosaurs. What about this DNA stuck in amber, though, I hear you say? That's what happens in Jurassic Park. A mosquito bites a dinosaur, drinks its blood, gets trapped in gooey resin. The resin hardens into amber, and 66 million years later, ta-da! Some dino DNA. So let's examine this directly. A typical female mosquito takes around 5 microliters of blood. That's 0.005 milliliters. Each microliter of vertebrate blood contains roughly 10,000 nucleated white cells, the only DNA source in mammals. Red cells in mammals are anucleate. Dinosaurs likely had bird-like nucleated red cells, which helps a bit, but the digestive enzymes in mosquitoes start degrading nucleic acids within hours, so the mosquito would need to be trapped in amber extremely quickly. Let's assume it was trapped very quickly. Amber preserves external morphology amazingly well. The mosquito itself looks amazing in the amber 66 million years later. But the internal tissues dehydrate and oxidize, degrading DNA. Micro cracks in the amber then let in oxygen and water, further accelerating the degradation process. And to top it all off, polymerized terpenes in amber are not a gentle storage medium for DNA. Amber gives us feathers, pollen, mites, even lizard toes in stunning detail. What it doesn't give us is intact dino genomes. Unfortunately, Jurassic amber is not a freezer. It's a resin coffin with chemicals that damage DNA, actually degrading it faster than it would outside of the amber. So, if not amber, how might we bring back a dinosaur? Well, there are 3 real possibilities. De-extinction strategy A, we edit a close living relative. This is the mammoth-elephant playbook, essentially. We could identify the differences between an extinct genome, like the mammoth, and a living creature, like an elephant, and then use CRISPR tools to edit living DNA of the elephant to create a mammoth-like creature. The Asian elephant has just over 3 billion base pairs. Millions of single-letter changes are required to change the elephant to the mammoth. We don't need them all, just the ones that change functional traits, things like hair growth to get the woolly, fat deposition, hemoglobin, oxygen efficiency, cold-tolerant cell membranes to ensure the mammoth can survive the cold temperatures. To do this, we need CRISPR-Cas9 tools. These cut and replace DNA. We need base editing tools. These perform single-letter swaps in DNA without cutting. And prime editing tools, which search and replace sections of DNA with fewer breaks. De-extinction strategy B, we resurrect traits inside living dinosaurs, aka birds. Birds are dinosaurs. Just as Dr. Grant tells us in Jurassic Park, all birds alive today are descended from the theropod lineage of the Cretaceous area. Chickens, for example, already hide dormant developmental programs for tails, teeth, and clawed digits. By adjusting early signalling pathways, researchers have produced chick embryos with snout-like premaxilla and tooth buds. A full Chickenosaurus would demand coordinated edits across dozens to hundreds of loci, plus careful embryology, but it's conceptually achievable. De-extinction strategy B, we clone dinosaurs from viable cells. Cloning by nuclear transfer, somatic cell nuclear transfers, is a laboratory process used to create a genetically identical copy of an organism. This requires an intact nucleus from a preserved cell. These exist for some recently extinct animals. For example, in 2003, the Pyrenean ibex, also called the bocado, was cloned from stored tissue. The ibex only survived 7 minutes due to lung defects. This brief but historic resurrection was the first de-extinction of a species that had previously become extinct. In 2020, a black-footed ferret was cloned from cells collected in 1988. Whilst not quite extinct yet, this added genetic diversity back into an endangered species. There are so-called frozen zoos being created. These are biobanks that hold thousands of cryopreserved cell lines across hundreds of species. These now exist, enabling the potential rescue of these species in the future, if they become extinct. So let's look at strategy A for resurrecting a dinosaur. How many edits of the DNA are enough to resurrect a dinosaur? In the case of a mammoth, you don't need a perfect mammoth genome to get a mammoth-like animal. If, say, 30 to 50 key gene networks control the thermal biology, the hair keratin, brown tissue, fat metabolism, cold-adapted hemoglobins, editing a few thousand sites might deliver a functional phenotype. This type of engineering isn't about perfection, it's about sufficiency. In the example of the extinct passenger pigeon, the last of which died out in 1914, there are already projects to resurrect these birds, which are mapping the differences between the current band-tailed pigeons and the extinct passenger pigeon DNA. Even if we match only the alleles for flocking, migration timing and diet, we might restore the ecological function of the extinct species, a sort of semi-passenger pigeon. Let's look at sequencing the DNA. As we said, the target coverage for a reference quality genome is 30 times. For a 3 gigabase genome, there are approximately 93 gigabytes of sequencing to do. A human-scale genome can be sequenced for under £1,000 in laboratory reagents. However, assembling a novel dinosaur genome with long reads and high C scaffolding could result in a DNA resurrection project cost of hundreds of thousands of pounds in scientist labour, quality control and computing power. And that's just for the sequencing of the dinosaur DNA. So let's assume we've managed to sequence the new DNA. What other problems are there? Well, Asian elephant gestation is about 22 months. A single generation therefore takes years. Large mammal de-extinction is necessarily slow therefore. How long is dinosaur gestation? Probably an average of around 6 months. So each iteration would be at least 6 months. No lab can reconstruct a complete dinosaur genome from the Mesozoic with today's chemistry. Remarkable soft tissue reports from fossils are just mineralized structures or degraded proteins. These are really useful for paleobiology, but not for cloning. You can't build a T-Rex from collagen snippets and hope CRISPR can just fill in the missing 3 billion letters. So the real science. There is only one real plausible path to resurrecting a dinosaur, and that's starting with a bird, or a living dinosaur, as it's also known. Editing hundreds of thousands of developmental switches on things like limb proportions, tail length, dentition, skin appendages, for example, and then accepting that the resulting animal is dino-inspired and not an actual dinosaur, not an actual genetic copy. That's the real science. Maybe not as cool as Jurassic Park, but still really cool. Let's assume that we actually managed to resurrect a dinosaur, by whatever means. What if we actually managed to hatch a real dinosaur? A 6 to 9 ton theropod would require hundreds of kilos of meat per week, reinforced enclosures, enriched habitats, and veterinary protocols we simply don't have. And they will have a behaviour that we can only guess at from bones. Where will these revived dinosaurs live? Their original ecosystems have shifted or vanished altogether. How will we give them social structure, enrichment, diets, disease control for Triassic fauna, let alone a fully grown Dilophosaurus? On an ethical note, every dollar spent reviving long-gone dinosaurs is a dollar not spent saving species now in freefall and near to extinction. There are over 40,000 species flagged on red lists in real danger of immediate extinction. So could we use these tools as a genetic toolkit for conservation instead? Could we restore species lost to the human destruction of habitats? Could we engineer disease resistance? For example, scientists are currently trying to help amphibians fight fungal diseases. Or could we help species with resilience to climate change? Scientists are currently helping corals to tolerate warm spikes, for example. Maybe we should aim for function instead of nostalgia. So, what's the real deal? What's the practical route for resurrecting a dinosaur? Step 1, pick a model and a target. A chicken and a compy, for example. Step 2, map trait modules, for example, tail length, forelimb digits, craniofacial patterning, skin appendages, scale feather balance, pigmentation. All of these things would need to be changed. Step 3, design edits. Dozens to hundreds of loci need editing, prioritizing regulatory elements that shift proportions without destroying the viability of the organism. Step 4, culture primordial germ cells and edit these in vitro. Step 5, create germline chimeras hatching surrogates that produce edited gametes. Step 6, breed these for combinatorial traits. This stacks edits across generations. And finally, step 7, validate the new traits, analysing gait analysis, bone morphology, behaviour, welfare, etc., to make sure that you actually have your targeted dinosaur. So, Jurassic Park has filmed a near-perfect genome from amber, a fleet of cloned Cretaceous giants, is not compatible with real chemistry or math. But de-extinction as a discipline is real, rigorous, and rapidly improving. We can resurrect genes, traits, and ecological functions, even whole species when cryopreserved cells exist. The future is not a park full of velociraptors, but the future could well be editing birds' DNA to explore evolution. It could be patching genetic holes in endangered species to prevent extinctions. And it could be engineering traits in current species to help them absorb things like climate shocks. That's less cinematic, until you realise it gives us something the movies never promised. A chance to use the past to fix the future. If you enjoyed this look into the real science of Jurassic Park, hit subscribe and I'll see you next time.